Hi, I'm Fred Kramer. I'm at the Public Health Research Institute here in Newark, New Jersey. And today I'm going to be talking about super-selective nucleic acid amplification primers. It's been a long-sought medical goal to detect extremely rare cancer cells in clinical samples that possess abundant normal cells. The trick to detecting the cancer cells is that they will contain mutations characteristic of the cancer cells and not found in the normal cells. And quite often these mutations are single-nucleotide polymorphisms present in the genomes of the cancer cells. The advantages of very early detection of rare cancer cells is that you can detect the presence of the cancer at a very early stage and treat it where the level of cancer cells is so low that successful outcomes are much more probable. An example that comes to mind is if people inherit genes which give them a propensity to develop cancer, such as the BRCA genes. Instead of, they give you this possibility of having cancer, breast cancer. It would be so much better if instead of having to do prophylactic surgery, for instance, the Angelina Jolie decided to remove her breasts rather than risk getting cancer. It'd be so much better if you could detect the cancer at such an early stage, long before any lesions actually appear in the breast, that you could then treat the cancer early. Also, if people have benign tumors that have the potential to metastasize, if you could very early on detect the changes that occur in those cells that allow them to metastasize, again, early treatment would be possible. And indeed, if you have been treated for cancer and a tumor has been removed or you've had a bone marrow transplant, there's always the possibility that cancer cells remain. This is the problem of detecting minimal residual disease. And current practice is quite often, because the minimal amount of cancer cells are not detected, is to still treat the patient with drugs to prevent the recurrence of cancer. But these drugs often have toxic side effects. How much better it would be if there was an extremely sensitive method for detecting cancer cells that would allow you to test the patient after the surgery and continue to test the patient in order to determine that clinical studies, of course, would have to be done first, but to check the levels of the residual cancer cells and only provide treatment if that level rises. You could then monitor patients over time. Similarly, if you're treating cancer with drugs and there's a possibility that mutations can occur in the cancer cells that cause those cells to become resistant to the drugs, how much better it would be if you could very early on detect those mutations and change the treatment regimen before serious problems arise. And lastly, even during the time that surgery is being carried out, quite often biopsies are done while the patient is still in the operating room to determine if additional tissues surrounding the tumor need to be removed. However, sometimes tissues are removed even when there's no evidence of cancer, because if cancer cells were in those tissues, it would have serious consequences. But if there were very rapid methods for determining the presence of rare cancer cells, these sensitive methods could show if there's no cancer cells and tissue could be spared. Spurring the efforts to detect rare cancer cells by virtue of their mutations is the realization that cancer cells divide rapidly, they're subject to apoptosis, and the DNA molecules in the cancer cells that die are broken down, fragmented, and end up in the blood circulation, in the plasma. However, if the cancer cell is rare or the mutation that you want to detect is rare, most of the DNA fragments in the plasma will be wild type, and the DNA fragments containing the mutation from cancer cells will be extremely rare. On the other hand, if you could detect these extremely rare fragments, it's so much better to have a blood sample taken, or even just the plasma removed and the DNA isolated and analyzed, than it is to take physical biopsies inside the body. So our efforts have been to develop a gene amplification assay that can directly detect rare DNA sequences containing relevant mutations, even when those rare DNA sequences are present in a sample containing a very large number of wild-type normal sequences. And the way that we've gone about it is in polymerase chain reaction assay. So there is a problem with detecting rare sequences in polymerase chain reaction assays. If the only difference between the mutant sequence and the wild type sequence is a single nucleotide polymorphism. And in the next slide, I can show you the problem. Here is two PCR assays run, one with 1 million mutant molecules and the other with 1 million wild type molecules as template. And you use primers that bind to the region that you wish to amplify. And one of those primers is designed to bind to the place in the sequence where the mutation will either be present or not. So that's shown in the upper left-hand corner of the graph there. That shows a primer, and a little circle represents a particular nucleotide that will be complementary to the nucleotide in the mutant sequence, but not complementary to the nucleotide in the wild-type template. And you see amplification occurs, exponential amplification, doubling every thermal cycle, every set of thermal cycles. And in these experiments, we use cyber green, something that lights up the resulting amplicons. And what you can see is there's really no difference in the amplification of the mutant gene and the wild-type gene. And the reason is, even though, let's say the primer was 20 nucleotides long, if 20 nucleotides are perfectly complementary to the mutant template, and 19 are perfectly complementary to the wild-type template, still both primers bind very well and make a product. So you really can't use conventional PCR primers to distinguish a mutant from a wild type. But what I'm going to be talking about today is a new design for PCR primers, which is highly selective. We call it SuperSelective primers. It's sort of shown schematically in the upper left-hand corner. When you replace one of the two primers, the forward primer, the primer that is going to bind to the place in the genome where there either is a mutant or not a mutant, when you use these primers, the mutant templates are amplified in the usual way. That's shown by the red curve that you see here. And you early on have amplicons being made indicative of the presence of those 1 million template molecules. But on the other hand, if exactly the same reaction is run, except instead you use 1 million wild-type templates, you see here that there is a great delay in the generation of amplicons from the wild-type templates. It's a 19-cycle delay. And this is because this unique design that I'm going to describe selectively discriminates against the wild-type template, even though the only difference is a single nucleotide polymorphism between the mutant and the wild-type. The next slide shows the design of these primers in greater detail. So the top sequence, the one that has a long red sequence of about 24 nucleotides, then a curved sequence, and then a green end sequence. That's the primer. Now, let me sort of describe how and why we design it this way. The starting point is that primers that are 20 nucleotides in length are far too long to make a difference between a mutant and wild type. That's the 19 versus 20. But on the other hand, if you could use a short primer, a primer that was, say, 7 nucleotides in length, then there's a substantial difference in the stability of the primer-target hybrid that's formed with a perfectly complementary primer sequence forming a 7 base pair hybrid, whereas the mismatched primer sequence on the wild type template makes a far less stable primer target hybrid and is much less likely to occur at the 60 °C annealing temperatures that are used in PCR assays. However, if you just used a primer that was only 7 nucleotides in length, it would bind to 100,000 different places in the human genome. It's not sufficiently specific to just bind to the region of the human genome where the mutation is that you want to amplify and look at. So what we've done is we've designed a primer that has 3 parts. The first part is labeled anchor sequence, and it's, in this case, 24 nucleotides long. And it is designed, it's very much like a regular primer in that it's so long that it only binds to the specific sequence very close to where the mutant nucleotide is located. This gives you the specificity for the primer to only go to the right place in the human genome that you want to study. Then on the other end, on the 3' end of the primer, you see a 7 nucleotide long, what we call it a foot sequence. And this is going to be the operating primer that either forms a perfectly complementary hybrid with the mutant you want to detect, or forms a mismatched hybrid with the wild type, and therefore is much less likely to form, less stable. And in order to separate the actions of the anchor sequence from the foot sequence, there's a connecting sequence called a bridge sequence. And it's completely artificial. It's chosen by us. It's chosen by us to not be complementary to the region of the template molecule that is labeled intervening sequence in the diagram. So when this hybrid binds, when this primer binds, the idea is that the foot sequence would have an independent action and would be highly selective with respect to binding to the mutant that you want to amplify. The only further thing I want to point out in this diagram is that the PCR conditions that we use are perfectly conventional. There's really no difference here from any other PCR. The only thing is we substitute for the forward conventional primer a forward super-selective primer. So the next diagram just shows the principle of operation here. The super-selective primers will bind to all of the templates, irrespective of whether they're wild type or mutant, via the anchor portion of the primer. Then they will be separated by the bridge sequence from the foot. And the foot will only bind if the key nucleotide, we call it the interrogating nucleotide, which in this primer shown in the diagram is a guanosine, a G. Only if the interrogated nucleotide is complementary to the C that's present in the mutant, will the foot of the primer anneal to the template. Whereas if there's a wild type, which is shown as an A here, the GA pair will not form, and the probability of the foot of that, the 3' foot of the primer binding to the template is much, much lower because there are less nucleotide pairs. There are less base pairs in the mismatched primer than in the matched primer. So there's a special aspect of understanding the operation of these primers. And I will come back to it again later, but it's shown here in this diagram. And this special aspect is that the selective step only occurs when the superselective primers bind to the template molecules that are originally in the sample being studied. So if you see in the top diagram of the three that are shown, the superselective primer is shown in red, and it's bound to the template shown in black. The anchor portion is separated from the foot portion by a sort of bubble, a non-base paired bubble formed by the bridge sequence of the anchor primer and the intervening sequence in the template. But because what we're showing here is the binding of the primer to the mutant, extension occurs and an amplicon is made. That's that dotted line in the top diagram going towards the right. So what happens in the next round of synthesis? And the next round of synthesis, the next cycle, the amplicon that's made, which is shown in red in the middle diagram, serves as a template for a conventional reverse primer, shown as the solid blue arrow in the middle diagram. And it is extended to make a complementary amplicon. But here's the beautiful thing. The complementary amplicon is a copy of the entire red original amplicon made from the superselective primer, and that is the bridge sequence of the superselective primer, which is completely arbitrary and chosen by us, becomes a part of the product amplicon, the dotted blue line in the middle diagram. As a consequence, in the second cycle after the amplicon was made, which is now we're down into the bottom diagram here, the blue complementary amplicon now serves as a template for the superselective primer to bind. But this time, there's no separation between the anchor portion of the primer and the foot portion of the primer. In fact, the total superselective primer, 45 nucleotides in length, acts as a perfectly conventional forward primer and binds to the amplicon. So the point is that once a superselective primer has copied an original template and made an amplicon, the resulting amplicons are synthesized very efficiently, just as in a normal PCR reaction. So all that we're counting on here to discriminate a mutant from a wild type is that the superselective primer, when it binds to the wild type, where there's a mismatched base pair, is far less likely to initiate the synthesis of an amplicon, from which there will then be efficient exponential amplification. So what I'd like to do is show you some of the experiments we've done with superselective primers, both to see how they operate and also to determine what is the best design of the superselective primers, and also how they work, what their mechanism of action is. I just want to say before I go into these slides, if you have questions, you can send them in while I'm talking. My colleague Salvatore Maris, who did this work with me, is monitoring those questions, and he can bring them in to me, and if it seems appropriate, we can answer them as we go. So please, by all means, do send in questions. Okay, so let me show you a set of reactions which were performed with superselective primers. Now, I think I should tell you a little something about the nomenclature here. So you see it says superselective primer 24-14/14-511. All that means is The anchor sequence is 24 nucleotides long. The bridge sequence is 14 nucleotides long. The opposing sequence in the template, the intervening sequence, is 14 nucleotides long. And the foot is 7 nucleotides long, of which 5 nucleotides are perfectly complementary to both the mutant and the wild type. The interrogating nucleotide is at the penultimate position from the 3' end, and therefore only matches the mutant sequence, but not the wild type sequence. And the 3' terminal nucleotide is perfectly complementary to both the mutant and the wild type. But we place the interrogating nucleotide at the next to the last position because we reason that if the next to the last position does not form a base pair, as is the case with a wild type template, the terminal 3' position will also not be able to form a base pair at 60-degree annealing temperature. So really you have the mutant hybrid would be 7 base pairs long, and the foot hybrid, and the wild type would only be 5, and it would have a non-bonded 3' end. Okay, most of the experiments I'm going to show you all use the very same mutant as a test object. It's the human epidermal growth factor receptor mutation L858R. It is characteristic of cancer cells, and when it's present, it tells the physician that that tumor is sensitive to drugs, antibodies that will be effective against the tumor, so it's important to measure it. But from our point of view, it's just a mutation. And these experiments were done with fragmented plasmid DNAs. The mutant DNA contains the mutant sequence, and the wild type DNA is absolutely identical except for one nucleotide, which is found in the wild type. And the way in which these experiments were done is we're showing you PCR reactions here. The amplicons are detected with cyber green that lights up the double helical amplicons that are made at the end of the chain elongation stage of each cycle. So what we did is we prepared 6 tubes, each of which contained 1 million wild type templates. But in each of those 6 tubes, there were different numbers of mutant templates. And you see that in the key on the right: 1 million, 100,000, 10,000, 1,000, 100, or even 10. 10 mutant molecules in the presence of 1 million wild types. We also put in a control that had no mutant molecules. That's the curve shown in green. And we even had a no template control, and you see nothing is made when there's no templates present. The key thing to see here is that the mutant alone, 1 million mutants, are so discriminated against by the use of these primers that the amplicons only begin to arise after about 40 cycles. Whereas because of the presence of the mutant templates, you get earlier arisal of the amplicons due to the copying of the mutant templates. And not only that, when they arise is a measure of how many mutants are present in the sample. This is a classic real-time PCR assay. The only thing that distinguishes it from a classic assay is one of the 2 primers is a SuperSelective primer. But other than that, there is this classic relationship between how many cycles it takes to make a significant number of amplicons and how many target molecules were present in the sample. So I'm going to show you how we analyze these. Here is the same slide, and I just put a dotted line there. The instrument we're using, Biorad IQ5, automatically analyzes the real-time results and determines a value above background, which is schematically shown by this dotted line. So for instance, the 1 million mutants that pass that dotted line something like after 23 cycles, Whereas if you look down at, say, the 100 mutants, that seemed to pass at around 36 cycles. So the idea is if you had less targets present, it takes longer before the exponential amplification process produces a result. To further show you how this is done, if you take those cycles at which the threshold or the key amount is passed, and you plot them against the logarithm of the number of mutant templates in the sample, you basically get a straight line which allows you to quantitate the number of mutants. The relationship is that the threshold cycle is inversely linearly proportional to the logarithm of the number of templates in the sample. This is a concept that our own laboratory introduced into the field in 1989 in an article in Nature and in Clinical Chemistry. But I think it's worth seeing that at log 1, which is 10 mutant molecules, the threshold cycle came up a bit earlier than one might expect from the others. It suggests that something else is being synthesized. So let me go further, and with the very same data, the dotted green line shows the threshold cycle determined with just 1 million wild types. And you see the threshold cycle for the wild type molecules is pretty close to the threshold cycle of 1 million wild type molecules plus 10 mutant molecules. And it suggests that discrimination is not perfect here. And so we decided to explore how we could increase the selectivity by redesigning the SuperSelective primers. And the first thing we wanted to explore was the size of the foot sequence, which in the illustration you have here is 511. It means 7 nucleotides long. But in the next slide, we repeated the experiment. The only difference is the primer now was 611, 8 nucleotides long. And this, you can actually see this makes the problem worse. There's the one with 10 mutants is not distinguishable from the sample that was pure wild type. So really, we wanted to explore making the foot shorter. And in the next slide, oh no, this is the current slide. In the next slide, you can see the results of those experiments. So this just shows the results with three otherwise completely identical reactions. The only difference is the SuperSelective primer had a different length foot. And so the green data, that's the 611 foot. You see that falls off. The 511 falls off just a little. But once you go to 411, you get a beautiful straight line detecting even 10 molecules of mutant in the presence of wild type. But there is what you also see is the shorter the foot, the later are the threshold cycles, the higher are the threshold cycles. And this is because at any given instant, at 60-degree annealing, the probability of forming a foot-target hybrid is really low. I mean, most of these at 60 degrees, something, say 6 long, is most of the time not hybridized, even to the mutant. All that this shows is that occasionally it does hybridize, or at any given instant, it's at equilibrium. A small fraction of the mutants are hybridized to the foot, and an even far lower fraction of the wild types, very much far lower fraction of the wild types are hybridized to a foot. And that's why that reaction, even with 1 million of them, is delayed so much that it doesn't interfere with the quantitation of the mutants in the sample. There's other things that we wanted to study. One was the effect of let me see if I have this here. Yes. Oh no, I'm sorry. Before I go on, we also studied the location of the interrogating nucleotide in the foot. And here you see in the upper left-hand corner are the results obtained. In each case, there's 1 million wild types or 1 million mutant templates. Yeah. Is that right? Yes. So the red is 1 million mutants and the green is 1 million wild types. And in the upper left is where the foot had the interrogating nucleotide at the 3' end. And just below that is the location at the penultimate position where all the previous work was done. And so forth. We moved the interrogating nucleotide farther and farther away from the 3' end, but left all the other characteristics of the primer the same. So the size of the bubble was the same, the size of the anchor sequence was the same. And what you see is when you get away from having the interrogating nucleotide 3' end, the discrimination is less good. But pretty much if the interrogating nucleotide is at the 3' end or the position next to the 3' end, you get very good discrimination. We were also concerned with the symmetry of the bubble. I mean, all of these experiments are done, all of these experiments are, well, wait a minute, no, I'm sorry, I don't think that's right. We were concerned with the size of the bubble next. I'm sorry. So here's an experiment where we tested 3 different primers, all containing the same foot, a 511 foot. But we used a shorter bridge sequence and arranged the anchor so that the result would be a shorter intervening sequence. So we could get bubbles of a different circumference. So our standard one was a 14-14 bubble, which really means 14 in the nucleotides in the bridge, 14 nucleotides in the intervening sequence, another 2 nucleotides in the circumference of the bubble generated by the anchor hybrid, and another 2 nucleotides generated by the foot hybrid. So that would be 14 plus 14 plus 4 for a circumference of 32 nucleotides. We also tested a shorter bubble, which is a bridge sequence of 10, intervening sequence of 10. That's a total circumference of 24, and a big bubble with 18 plus 4, that's 40 nucleotides. And did exactly the same experiments with different numbers of mutant templates in the presence of 1 million wild types. And what you can plainly see here is the bigger the bubble, the more linear the results. The more, or rather, the less interference you get from the wild type sequence, the greater is the discrimination by using bigger bubbles. And yet, the bigger the bubble, the later is the threshold cycle. And the reason is the formation of anything thermodynamically is based on 2 factors. One is the strength, the enthalpy of the hybrid formed by the foot and the target of the foot. But the other factor is the entropy, the probability that the foot and the target can get together. And the bigger the bubble, the less likely it is that the foot and the target will find each other. And once they fall apart, less probable they'll come back. So at any given instant, at equilibrium, there are far less hybrids that occur, but yet you get the best discrimination by lowering the probability of formation. And I'll go into this more. And there was a last question that we had: what happens if we change the symmetry of the bubble? What if we make a big bridge sequence and a shorter intervening sequence, or vice versa? And just to make a long story short, it seemed not to matter. In this case, we had 5 different primers, all of which created bubbles of the same circumference, but with different contributions in length from the bridge sequence and from the intervening sequence. And it seemed not to matter. All that mattered was, in effect, the entropy factor, the size of the bubble. Okay. Okay, and some questions have come in, but I'm going to handle them at the end. Now, so we've seen that a bigger bubble and a shorter foot makes for better discrimination. So here's an experiment, everything else being the same, in which we combined the bigger bubble, an 18-18 bubble. It has 18 in the bridge, 18 in the intervening sequence, and the shorter foot, 411. And you can see the beautiful results that you get. The 1 million wild types, the dotted line on the left-hand panel there, is very far removed from the threshold cycles that you get when there is mutant present in the sample. So this allows you to measure, based on the threshold cycle, the amount of mutant in the sample, despite the presence of the mutation. Okay, let's look what else we have here. So all of these experiments were done with plasmid DNA. Now we switched over to see if this works with human genomic DNA. And we were able to obtain a cell line, H1975 cell line, that contained the same EGFR mutation. And we also were able to obtain cells that were wild type. And we isolated the DNA from each of these and fragmented it in the same way and repeated this test. And you can see that, again, even though the entire genomic DNA is there, you get this beautiful linear relationship between the log of the number of mutants targets and the threshold cycle that is measured in real-time PCR reaction. So all of these experiments were done with this particular mutant. We've actually done experiments with a number of different mutants, with different nucleotides being the variable nucleotide. And pretty much they all work. The only exception, we got slightly less good results if the mismatched foot and wild type target was a GT base pair. Then we just simply used the other strand as a template and had no trouble. But here's an example of a totally different mutant. This is the BRAF V600E mutant. It's a marker of cancer that causes colorectal cancer. But I mean, that's not important. But what distinguishes this particular mutation is it occurs in an AT-rich region. That is, the target sequence is AT-rich, and therefore the foot sequence is also AT-rich, as opposed to the EGFR L858 mutation, which was GC-rich. And what we found here is, again, we got beautiful results. But this time we only needed a smaller bubble and a slightly longer foot to get those good results. And the reason is the AT richness of the target sequence played the same role of making the probability of forming an initial foot target hybrid lower, and that leads to this selectivity. Okay, let's see. I want to find the next one here. Let's see. No, that we showed. This we showed. Okay. One further point is, no, this is the one I just showed. I'm sorry. One further point is that we did a comparison experiment using samples containing mixtures of mutant targets and wild-type targets. We compared the results that we got with those samples based on the threshold cycles that were determined. And the same samples were analyzed by digital PCR to see how many mutant targets were in there. And you see there's a beautiful correlation between direct detection of the number of targets and the threshold cycle that's determined by classic real-time PCR reaction. So we feel that the substitution of these Kramers solves the problem of selectively amplifying mutants that you want to detect. And one of the most beautiful things is that perhaps this can be done with a liquid biopsy, just taking DNA from blood plasma and looking for these rare mutants is a very, very early indication of the presence of cancer cells. So I wanted to discuss the mechanism by which these work. Okay. So first of all, as I've mentioned, there's a very low proportion of foot target hybrids formed, and the mismatched foot target hybrids with the wild type are thermodynamically much less likely to form and initiate an amplicon that can then be efficiently amplified because the foot target hybrid is shorter and less strong. But it's hard to imagine that thermodynamic difference in stability of the hybrid, 7 long versus 5 long, let's say, is enough to account for 19 or 20 cycles of discrimination, of repression of the wild type. So what we speculate is that there's also a kinetic aspect to the determination of whether amplicons are made from mutants or wild type. You know, classic PCR primers, say 20, 24 nucleotides long. Once they form, their mean persistence time is measured in minutes. So effectively, when they form, they stay formed. That's because they're so long, and there's so many base pairs that glue the primer to the target. But with the foot primer, with something that's 5 or 7 long, the mean persistence time of those short, weak hybrids is measured in milliseconds. Not only that, the perfectly complementary hybrid with the mutant sequence is going to have a persistence time that's much longer than the persistence time of this itty-bitty hybrid formed with the wild type. So what we're speculating here is that since the hybrids only exist for a short amount of time, the probability that they'll be able to interact with a DNA polymerase and generate an amplicon, it also depends upon how long they exist. They have to move around the reaction. They have to move within the annealing solution, encounter a polymerase, and form an effective functional hybrid. And they have to do that before they fall apart. And since the wild type, shorter, mismatched hybrids exist for a shorter length of time, the probability that they are going to form an effective complex with DNA polymerase is lower. So you have two factors there. You have the factor of the thermodynamic abundance of a matched hybrid, a perfectly complementary hybrid versus a mismatched hybrid. And you also have the factor that the mismatched hybrids are weaker and exist for a shorter amount of time. And it's the combination of both of these factors that lead to the extraordinary discrimination. And there is some evidence for that in our results. Let me go back to where we studied bubble, the effect of changing the size of the bubble. Changing the size of the bubble should definitely affect the overall abundance of the foot target hybrids. But it should have the same effect on the mismatched wild type hybrids as it does on the perfectly complementary mutant hybrids. The relative abundance of those two should only be determined by the enthalpy, by the energy that's saved by forming the hybrid. And yet, we got better results the bigger the circumference of the bubble. But from a kinetic point of view, these bubbles are single strands connecting the anchor hybrid to the foot hybrid in the annealing solution, and the water molecules in the annealing solution are bouncing around chaotically and exerting a force on the single-stranded bubble. And the bigger that bubble, the greater the force. So bigger bubbles tend to rip apart the foot target hybrids. And because the perfectly complementary mutant foot target hybrids are stronger than the weaker mismatched foot wild type hybrids, we feel that the wild type hybrids are ripped apart more readily by the action, random action of the molecules in the solution on the bubble. So the bubble is playing a definite role here in terms of enhancing discrimination. Okay, let me show you pictures of my colleagues who did this work. This is my colleague Salvatore Maras. He invented the concept of superselective primers, and he's also monitoring your questions right now. Here is Diana Vargas, who carried out all of the experiments that we've shown today. She is our research associate in our laboratory. And here is Sanjay Tyagi, who Sanjay and I have shared a lab for 27 years here at the Public Health Research Institute. Is that right, 27? Long time. Let's see. No, even longer, but okay. Okay. So now, let me put one slide on the screen, which I am required to, is that you can obtain continuing education credits by signing on to this website for watching this talk. And now I'm going to take some of the questions that have been sent and see if I can answer them. So let me put a picture of this lanternfish back up. I like it. So the question is, have we created software for designing these unique primers? And the answer is no, we have not yet. But we do have, we have a feeling for how to go about designing them. We have submitted a manuscript, which we hope will be accepted and which describes what to do. And I think soon after that, there will be programs to design these primers for sure. In the meantime, you definitely can send queries on design to our laboratory. And I think the best person to send it to is Dr. Maras. And if you, I will tell you his email address if you have a pen, and you can contact him directly. So it's Maras SA, so MARRASSA at Rutgers.edu. Rutgers is spelled RUTGERS, and he can help you design these primers. We had a question about whether we tried these primers with digital PCR, and the answer is yes, we have. And of course, in digital PCR, you want to distinguish between the much more abundant wild types and the mutants that can end up in any given well. But in our preliminary, the way digital PCR works is there's basically going to be one mutant per well to initiate the PCR reaction, as and many wells won't have a mutant at all. Under that situation, where there's only one template, and you've already seen how rare it is to form a foot target hybrid, there's sort of a Poisson situation in terms of when the reaction starts. With 10, there's no problem. But when you get down to 1, there's some variability in the different wells that start with one molecule. So what we have to do there is design less selective primers, which will not have that Poisson problem. We have not done that yet, but it's certainly on our agenda to do it. And of course, you can try it out there. You can have longer foot sequence, shorter bubble sequence. That should be less selective, and perhaps then initiation, even when there's only one molecule, will occur at a quite predictable cycle. At least that's what I hope. The last question is, can you design superselective primers if you don't know the specific mutation that exists? And the answer to that is no. I think you need to know what it is you're looking for. Let me just say as last point, one of the beauties of being able to do a classic PCR reaction with superselective primers is that it opens up the opportunity for the design of multiplex superselective primer assays. That is, instead of looking just for one mutation, you can look for a series of different mutations, each of which is relevant, for instance, for cancer. It can tell you what drugs will work. It'll tell you what stage the cancer is at. It'll tell you something about where it's located, even though you might be looking at a liquid biopsy from plasma. In fact, it's so sensitive and so quantitative, and quantitative is the key, that for each patient sample, you should be able to get a sort of personalized picture of that person's situation, and therefore therapy can be adjusted and calibrated to fit that person's particular situation. And this might make for much better outcomes, not only because detection is early, but because multiplex detection of a series of significant mutations allows you to personalize and adjust the treatment. A special aspect of superselective primers is that the bridge sequence that we put into the primer is completely arbitrary. It's chosen by us. It's chosen so it's not complementary to the target, certainly. It's chosen so it's not complementary to any place in the human genome. It's chosen so it does not form hairpin structures, which would shorten the size of the bubble. But here's the lovely thing about it. Recall that the superselective primer becomes incorporated into the amplicon. And so the bridge sequence becomes incorporated into the amplicon, and the complement to the bridge sequence is incorporated into the complementary amplicon. And this represents a unique target for a probe, for instance, a molecular beacon probe, which our lab has invented some years ago, to light up the amplicon that was initiated with that primer. And so a whole series of different primers can be used, each specific for a different mutant target sequence, and each of them containing a totally different bridge sequence, and therefore a whole set of molecular beacons, each specific for a different complement to the bridge sequence, can be used, each labeled with a different colored fluorophore. So you should be able to do classic multiplex PCR assays against a series of targets, all of which are relevant to one particular clinical situation. And we're working on that now, and we're happy to work with other people who are interested in working on it. I think at that point I've pretty much said everything. I will ask my colleague Salvatore if there are any more questions, and if not, we will sign off. I very much thank you all for watching and hope you found it interesting. We'll see what Salvatore has. There are no more questions, so I think we're done with the presentation. I want to remind you that you can get CME credit, and also if you have questions, you can send an email to Salvatore or to me. I'm Kramer, FR, KRAMER, FR, @rutgers.edu, and we'll be happy to answer. Thank you, and I guess we'll sign off. I'm not so sure how we do that, but thank you.